Imagine spending like 9 years of your life and up to maybe $750 million to develop this revolutionary new drug.
Yeah, it's a massive investment of time and capital.
Right. And you run it through every established preclinical test. You give it to animal models up to and including non-human primates.
Standard battery of tests.
Exactly. And the data comes back absolutely clean. Like, it looks completely safe. You have every reason to believe you are about to, cure a disease and completely change the world.
You're popping champagne in the lab.
Yeah. And then the very moment it enters human trials, it just fails spectacularly. I mean, it causes severe life-threatening reactions. All that time, all that money, all that hope, just gone.
It is a devastating scenario.
Mm-hmm.
And when we look at the historical data, the core statistic here is genuinely staggering.
Oh, it's wild.
Over 90% of drugs that appear completely safe and effective in animal models end up failing in human trials.
90%.
90%.
Mm-hmm.
I mean, they failed primarily due to unexpected safety issues that the animal models just didn't catch, or simply because they aren't effective in human biology at all.
That is just a jaw-dropping failure rate. I mean, if 90% of the airplanes we designed looked great in the wind tunnel but crashed on their first real flight, we would completely rethink how we design airplanes, right?
Oh, absolutely. The whole industry would stop.
And that is exactly what we are exploring in today's deep dive. We are looking at this massive structural paradigm shift in medicine. We're moving away from traditional animal testing and entering the era of names. That's new approach methodologies.
Yeah, this is a fundamental reorganization of the entire biomedical research enterprise. We are shifting from a century-old reliance on animal models to this highly advanced, truly human-centric framework.
And to guide us through this, we're synthesizing a literal mountain of recent data today.
So much data.
Yeah, we're looking at the FDA's newly issued March 2026 draft guidance on names, the NIH's $150 million complement ARI program announcements, the latest Tox21 consortium data.
Right, the Tox21 stuff is fascinating.
It really is. Plus, we've got industry perspectives from biotech giants like Thermo Fisher and these really deep academic reviews specifically focusing on oncology.
And the breadth of these developments is really the most critical takeaway here. This isn't just an academic theory anymore.
No, it's happening right now.
Exactly. The regulators, the massive federal funders, and the biotech corporations, they are all shifting massive amounts of capital and policy in the exact same direction at the exact same time.
So whether you are a biotech investor trying to spot the next big technological leap, or maybe a medical professional keeping up with how therapies are approved, or just an intensely curious learner who wants to know how the medicines in your bathroom cabinet are being radically redesigned, this deep dive will unpack exactly how the future of drug discovery is being built right now.
It's going to change everything about how we approach medicine.
Okay, let's unpack this. Because to understand why these new approach methodologies are taking over, we have to look at the fundamental foundational flaw in the century-old animal testing model.
The root of the problem.
Right. It boils down to a simple truth with really complex biological consequences, which is animals are not humans.
The biological mismatch is, it's profound.
Yeah.
For decades, the pharmaceutical industry and regulatory bodies basically operated under this paradigm where animal testing was the absolute default requirement for gaining safety information.
But a mouse is not just a tiny human.
Exactly. Even a non-human primate, as close as they are to us evolutionarily, has entirely different metabolic pathways, different immune responses, and different genetic expressions.
And there is a chilling specific example of this from our sources, the drug TGN-1412. Walk us through the biology of what actually happened there, because it's pretty scary.
TGN-1412 is a classic tragic case study in the limitations of these animal models. This was an immunomodulatory drug, a monoclonal antibody, designed to treat diseases like leukemia and rheumatoid arthritis.
Okay.
Its mechanism of action was to bind to a specific receptor on T cells called CD28.
Right, the CD28 receptor.
Yeah. And during preclinical testing, it was given to cynomolgus macaque monkeys. The drug appeared perfectly safe.
No side effects?
No red flags whatsoever. But when it was administered to healthy human volunteers in a phase 1 clinical trial, the results were catastrophic.
What was the actual physiological reaction in the humans? Like, what happened to them?
Within minutes to hours, the human volunteers experienced this massive cytokine storm.
Oh, wow.
Yeah, their immune system just went into hyperdrive, literally attacking their own organs. They suffered multi-organ failure and required intensive care,
Just from a phase 1 dose,
just from the initial dose. And the reason this happened comes down to a microscopic biological difference.
which the monkeys didn't have.
Right. In humans, the CD28 receptor is heavily expressed on memory T cells, which act as aggressive responders. But in macaques, CD28 is expressed differently. Their memory T cells actually downregulate that receptor.
Wait, so the receptor is basically hidden in the monkeys.
Basically, yeah. So the drug bound to the macaque cells and just gave them a mild immune boost. But it bound to the human cells and triggered a biological nuclear explosion.
That is terrifying.
The animal model was structurally blind to the specific human immune pathway the drug interacted with.
And beyond the human cost, which is obviously the most important thing, there is a staggering economic toll to this flawed system. I mean, developing monoclonal antibodies, which are some of the most advanced and effective drugs we have right now, that can cost between $650 and $750 million.
It's an astronomical sum.
And it takes up to 9 years.
And a massive chunk of that cost and time is eaten up by these structurally flawed animal studies.
Yeah, the sources note that the cost of non-human primate testing alone is up to $50,000 per animal.
50 grand for one animal.
Right. So when running large preclinical trials, those numbers scale up incredibly fast. You are spending tens of millions of dollars on a testing platform that, statistically speaking, has a 90% chance of giving you the wrong answer about human efficacy.
It's just bad math. And alongside the scientific and economic pressures, we're seeing a massive ethical and social shift.
Which brings us to the 3 Rs, right?
Yes, the foundational concept of the 3 Rs. Replace, reduce, and refine.
Break those down for us.
Sure. The goal is to replace animal use where possible with scientifically valid non-animal methodologies. Then, reduce the number of animals needed by adopting more efficient approaches. And finally, refine practices to minimize stress and improve welfare when animal use is still strictly necessary.
And public opinion is heavily driving this. I mean, the data shows over 85% of U.S. adults now support moving away from animal testing to more modern, human-relevant methods.
Just a huge majority.
It's a rare issue where, ethical concerns, economic incentives, and scientific accuracy all point to the exact same solution. Let's look at it this way. Relying on animal models to predict human biology is like using a map of Chicago to navigate New York City.
Oh, I like that analogy.
Yeah, like the streets might look similar, there are tall buildings, there's a river, but if you follow the directions, your destinations are going to be completely wrong. You're going to end up totally lost.
That analogy holds up perfectly when you look at the granular details. I mean, you might recognize the general shape of the mammalian organ system, right?
A heart is a heart, a liver is a liver.
Exactly. But the specific biological intersections, the receptor binding affinities, the metabolic half-lives, they are totally different between a dog and a human.
But here's my question. If the failure rate is over 90%, and we've known about these biological mismatches for decades, why has it taken so long to change the map? Why are we only having this massive paradigm shift now in the 2020s?
If we connect this to the bigger picture, the delay hasn't been because scientists loved the old map.
Right.
It's been due to 2 massive hurdles: technological limitations and cultural inertia. For a long time, we simply didn't have a better map.
We were stuck with Chicago.
We were stuck. You couldn't just drop animal testing because the alternative was using nothing, which would mean testing completely unknown, potentially lethal chemicals directly on humans.
Which we obviously can't do.
Right. We haven't had viable, scientifically robust replacements until the recent exponential explosion of biomedical technology.
And that brings us to the actual tools being built to replace that old map. We really need to look deeply into what exactly constitutes a name. Because the term new approach methodologies sounds like, I don't know, a broad piece of corporate jargon.
It does sound like jargon, yeah. But it is a broad umbrella term, and deliberately so.
How does the government define it?
According to the Interagency Coordinating Committee on the Validation of Alternative Methods, that's ICCVM NAMs, encompass any technology, methodology, approach, or combination thereof that provides information on chemical hazard and risk assessment while supporting the 3 Rs.
Okay, so in plain English.
In plain English, they are advanced laboratory or computer-based testing approaches intended to model human biology much more accurately than an animal can.
Got it. Since our mission today is to truly understand the mechanics of this shift, we need to break open this toolbox. There are 4 main categories of NAMs we need to unpack. Let's start with the first one. In vitro methods.
the lab-based stuff.
Right. Now, I know in vitro literally translates to in glass, like Petri dishes, but we aren't just swabbing some cells on a flat piece of glass anymore, are we?
Oh no, we've moved far beyond flat Petri dishes. Traditional 2D cell cultures are still used for, like, basic toxicity screening. But the frontier of in vitro NAMs has moved into 3 dimensions.
3D biology.
Exactly. We're about 3D cell cultures, patient-derived organoids, PDOs, and microphysiological systems, which are often called organs on a chip.
I want to pause on organs on a chip because the engineering behind them is just fascinating. What exactly is a microphysiological system?
Well, to visualize it, picture a clear, flexible polymer chip, roughly the size of a USB flash drive.
Okay. Pretty small.
Very small. Usually it's made from a silicone-based polymer called PDMS. Inside this chip are hollow, microscopic fluid channels lined with actual living human cells.
Wow.
But it's not a static environment. Researchers physically pump fluid, like a nutrient-rich media or even human blood plasma, through these channels
Why do they pump it?
to mimic the mechanical shear stress of blood flowing through a capillary.
So you are recreating the physics of the human body, not just the biology.
That physical recreation is paramount. Cells behave completely differently when they are flat in a dish versus when they are experiencing the mechanical forces and 3D architecture of an actual human organ.
That makes sense. They need to feel like they're in a body.
Exactly. Take a lung on a chip, for example.
Okay.
The chip has a porous membrane running down the middle. On the top half of the membrane, researchers culture human lung epithelial cells and expose them to air. On the bottom half, they culture human endothelial cells, blood vessel cells, and flow a blood surrogate through it.
So you have air on top, blood on the bottom.
Right. And furthermore, they apply a vacuum to the side channels of the polymer chip, causing the membrane to stretch and relax rhythmically.
Wait, it's physically breathing.
It is mechanically simulating the respiration of a human lung.
That is wild.
It really is. This allows researchers to introduce an aerosolized drug into the top air channel and watch in real time as the drug crosses the lung tissue barrier into the bottom blood channel.
So we can see how human tissue metabolizes an inhaled drug without needing a human patient.
Precisely.
That leads perfectly into the second category in the toolbox. In silico, this is computational.
Yes. In silico refers to mathematical modeling, computer simulations, and increasingly artificial intelligence and machine learning.
Here's where it gets really interesting. How does a computer program test a physical drug? Like, it doesn't have a microfluidic channel to push chemicals through.
It tests a drug by analyzing massive, incomprehensibly large data sets. We are talking about multi-omics data.
Multi-omics, what does that cover?
That encompasses genomics, which is the DNA blueprint, transcriptomics, which is how that DNA is read into RNA, proteomics, which looks at the actual proteins built from that RNA, and metabolomics.
Metabolomics.
Yeah, that measures the chemical exhaust produced when cells function.
Ah, okay.
So an AI model can ingest all of this multi-omics data from thousands of previous human experiments.
So it essentially knows the baseline of how human biology works at a molecular level.
It knows the baseline and it understands molecular geometry. This involves something called QSR, quantitative structure-activity relationship models.
QSAR.
Right. An AI can look at the 3D chemical structure of a brand new unsynthesized drug, cross-reference it with millions of data points on how similar structures interact with human proteins, and simulate the toxicity pathways before a physical test even happens.
Before the drug even exists in the real world.
Exactly. You can test 1 million virtual compounds against a virtual human liver in the time it takes to set up one physical experiment.
It's like running 1 million flight simulations, testing every aerodynamic variable before you even bother fabricating the physical prototype of the airplane.
That's the perfect way to look at it. And the AI isn't just looking for basic toxicity. It's identifying complex, nonlinear patterns across the proteome that a human researcher might never spot.
So we have in vitro, the living chips, and in silico, the computational models. The third category is in chemico. Chemical-based. What does that mean in practice?
In chemical methods are completely cell-free experiments. They focus entirely on the direct biochemical interactions between a chemical and isolated biological molecules.
Give me an example.
A great example is testing for skin sensitization.
Like testing if a new cosmetic or chemical will cause a severe allergic rash.
Historically, that involved shaving an animal's skin and applying the chemical to observe the reaction.
Right, the rabbit test.
Yes.
Yeah.
But with in chemico methods, we understand the underlying molecular mechanism. Skin sensitization usually starts when a chemical acts as a hapten. Yeah, it means it penetrates the skin and binds covalently to specific skin proteins, altering their shape. That altered protein is what triggers the human immune system to react.
So instead of testing the entire immune reaction on an animal, you just test the first domino falling.
You isolate these specific synthetic proteins in a test tube and add the chemical. This is called a direct peptide reactivity assay.
PPRA.
Right? If the new chemical reacts with and binds to these isolated proteins, we know mathematically it is highly likely to trigger that immune cascade in human skin. No cells, no animals, just pure biochemical binding logic.
That is so elegant. And the fourth category involves lower organisms, which, well, it seems like a bit of a hybrid approach.
It is a hybrid. While the ultimate goal is fully human-based systems using lower organisms like C. elegans, a microscopic roundworm or zebrafish embryos is a massive step up in efficiency and a major reduction in mammalian testing.
Why zebrafish?
Zebrafish are surprisingly relevant. They are vertebrates, and they share about 70% of their genes with humans.
Oh, wow, I didn't know that.
Yeah. And more importantly, their embryos develop entirely outside the mother, and they are completely transparent.
Wait, transparent. So you can physically watch their internal organs form under a microscope in real time.
Exactly. Because of that transparency and their rapid reproduction, you can use them for high-throughput screening.
How does that work?
You can array thousands of zebrafish embryos in microscopic wells, expose them to a massive library of environmental chemicals, and computationally track any developmental signals, heart rate changes, or genetic mutations very rapidly.
So it's still animals, but way faster and more humane than using mammals.
It's a refinement and reduction strategy that provides excellent early-stage hazard identification.
Okay, so we have these four distinct tools, but the real magic happens when you combine them. The sources talk about this concept of the NAMS box. The academic reviews compare NAMS to a Lego factory.
That modularity is the entire point.
Right.
Just as you can build a landscape, an animal, a spaceship, or a skyscraper out of different Lego bricks, researchers don't just rely on one single NA.
They mix and match.
They do. They can combine the in vitro living chips, the in silico AI models, and the in chemico protein assays to build a custom NAMS box, specifically designed to evaluate one unique disease or one specific drug pipeline.
So if you want to test a drug for liver toxicity, you build a liver-centric NMS box. If you want to test an inhaled asthma drug, you build a lung-centric NMEMS box with breathing mechanics and immune cells.
Custom built every time.
These aren't just replacements for animals, they are fundamental upgrades. We are engineering the exact testing environment we need rather than hoping a mouse represents it.
What's fascinating here is that this modularity completely changes the philosophy of testing. We are shifting from a generic, one-size-fits-all animal model to a bespoke, engineered biological simulation.
But having a highly advanced Lego box of technologies is great in an academic lab, like it gets researchers published, but it means absolutely nothing if the federal regulators, the people who actually approve the drugs and allow them to go to market, don't accept the data.
The ultimate bottleneck.
Which brings us to the regulatory revolution.
This is where the tectonic plates of the industry are actively shifting. For almost a century, the regulatory framework explicitly demanded animal data. The 1938 Food, Drug, and Cosmetic Act essentially locked animal testing into the law.
That was largely a reaction to the elixir sulfanilamide tragedy, right? Where an untested drug formulation poisoned over 100 people.
Exactly. The government mandated animal testing to ensure that never happened again.
And that 1938 mandate made sense given the scientific tools available at the time, but that legal requirement remained in place until the FDA Modernization Act 2.0 passed in late 2022.
That legislation was a watershed moment.
What exactly did it change?
It formally removed the century-old statutory requirement for animal studies in the preclinical evaluation for investigational new drugs, or INDs, and biosimilars. Congress essentially gave the FDA the legal authority to say, if you have a better non-animal method, we can accept it.
But changing a law is one thing. Changing the massive bureaucratic machinery and risk assessment culture of the FDA is another.
Absolutely.
And that is why the March 18th, 2026 FDA draft guidance is such a massive deal. The document is titled General Considerations for the Use of New Approach Methodologies in Drug Development.
This guidance is the FDA giving the industry the actual playbook. They are communicating, we want you to use these tools, and here's exactly how you must validate them so our reviewers can trust the data.
Let's dig into the 4 core validation tenets from this 2026 guidance, because this is the rubric every biotech company is now being graded on. Tenet number 1, context of use, or COU.
Context of use is arguably the most critical step. It means defining precisely what the NAM is meant to do.
So you can't just throw a chip at them and say, approve this.
No, you cannot hand the FDA a lung and a chip and say, here, this replaces dogs. You have to define the biological boundaries.
What does that look like in practice?
You have to say, this specific microphysiological system is intended to evaluate radiation-induced vascular toxicity in a 3D lung tumor microenvironment over a 14-day period. You declare exactly what the tool can predict and implicitly what it cannot.
You declare exactly which Lego structure you built and what specific question it answers.
Precisely.
Tenet number 2 is human biological relevance.
You have to prove scientifically that the pathways and mechanisms your ink name is measuring actually mimic human biology.
So the math has to check out biologically.
Right. If your AI model predicts a toxic reaction in the liver, you must show that the mathematical logic maps onto known human physiological processes, like cytochrome P450 enzyme metabolism. The mechanism must be undeniably human.
Tenet number 3 is technical characterization.
This focuses on the operational engineering. Is the test reliable? Is it robust? Is it reproducible?
Like, does it work the same way twice?
Exactly.
Yeah.
If a pharmaceutical company runs a chemical through a liver on a chip in a lab in Boston and then runs it again in a lab in Tokyo, do they get the exact same metabolic readout? You have to establish scientific confidence through rigorous benchmarking against known reference chemicals.
And the final tenet, fit for purpose.
This simply asks the pragmatic question, does this NAM actually help the FDA make a regulatory decision?
Is it useful?
Right. Does it provide data that allows a reviewer to say, yes, based on this readout, this drug is reasonably safe to proceed to phase 1 human trials? The data must be actionable within the regulatory framework.
Now, I noticed a really surprising detail in the text while reviewing this FDA guidance. I had to read it twice. The FDA explicitly states that a NAM doesn't necessarily need to be fully validated to be used in a regulatory submission. It just needs to be fit for purpose.
That's right.
How can regulators accept unvalidated data? I mean, that seems like a massive risk.
It sounds contradictory on the surface, but this flexibility is actually regulatory genius.
How so?
Traditional full validation of a new testing method can take a decade. It requires massive multinational ring trials involving dozens of independent labs. If the FDA demanded full validation for every new AI model or every iteration of a lung chip, innovation would grind to a complete halt.
Ah, okay, so it would be too slow.
Way too slow. Instead, they are adopting a weight of evidence approach.
So they are looking at the aggregate picture rather than just one standalone test.
They look at the totality of the data. If a drug developer has a specific, urgent toxicological concern, say a specific type of kidney toxicity, and they use a highly characterized but perhaps not universally validated NAM to address that specific concern, the FDA can evaluate it.
In that specific context.
Exactly. If that NAM data aligns with other known pharmacologic data and computational predictions, the FDA can accept it for that specific context of use. It allows for the early adoption of cutting-edge tools, speeding up innovation while maintaining a strict safety net through a holistic review.
The FDA has built specific pathways to help companies get these tools approved, too. They have the ISTAN program and the DDT program.
ISTAN stands for Innovative Science and Technology Approaches for New Drugs. It's a pilot program designed to encourage early engagement.
So companies can talk to them before they submit.
Yes. A company can approach the FDA with a completely novel piece of tech, and the FDA will work collaboratively with them to design the validation plan.
And what about DDT?
The Drug Development Tool, or DDT, qualification program goes a step further. Once a tool, like a specific liver on a chip, is formally qualified through this program, drug developers can use it in multiple different drug applications without having to reprove that the tool itself works every single time. It becomes a standardized part of the accepted regulatory toolkit.
Okay, so the regulators have given the green light, the laws are changed, the guidance is published, the pathways are clear, but transitioning an entire global industry from animal cages to microfluidic chips and supercomputers requires massive capital and immense infrastructure.
Oh, absolutely.
Where is the money coming from to build this new map?
This brings us to the massive federal mega shift. And it starts with understanding the scale of the data required. You cannot train an AI to predict human toxicity without giving it a mountain of biological data first.
Right, need the baseline.
That's where the Tox 21 consortium comes in.
Tox 21. Toxicology in the 21st century. This is a massive collaboration between the NIH, the FDA, and the EPA. And the physical infrastructure here is incredible. They operate at the National Center for Advancing Translational Sciences, or NCNTS.
The NCNTS facility utilizes high-throughput robotic screening systems. They're using 1,536 well plates.
1,500 tiny little wells on one plate.
Yeah, and they use acoustic dispensing, literally using sound waves to move nanoliters of fluid with extreme precision to test a massive chemical library called the Tox2110K library.
That library is 10,000 environmental chemicals and approved drugs, and the robotic arms are testing these chemicals across over 100 different biochemical assays day and night.
They have generated over 150 million data points.
150 million.
They are mapping exactly how every one of these chemicals interacts with human cellular pathways. To put that in perspective, the EPA uses this specific TOX21 data to look at estrogen receptor activity.
How does that help the EPA?
They can computationally predict if a new industrial chemical will act as an endocrine disruptor, interfering with human hormones, and use that data to prioritize it for stricter regulatory testing.
150 million data points. They are effectively mapping the human-chemical interaction universe. But TOX21 has been building that database for a while. The groundbreaking news we are focusing on is the Complementary Eye program.
The NIH Common Fund's Complementary Eye program is the financial rocket fuel for this current transition.
Tell me about the money.
The NIH recently announced a massive $150 million total investment specifically to develop, standardize, and scale EMS across the United States.
Let's break down where that $150 million is actually going. First, there is a massive $25 million grant awarded to NYU Langone Health and Sage Bionetworks to build a NEMS data hub and coordinating center.
That's a huge piece of the puzzle.
But why do they need $25 million just for a data hub?
Because standardizing biological data is incredibly difficult.
Oh, because everyone formats it differently.
Exactly. If you have hundreds of researchers generating complex 3D organoid imaging data, transcriptomic readouts, and AI simulations, but they are storing it in completely different proprietary formats, the broader scientific community can't use it.
It's locked in silos.
Right. The hub will standardize this highly heterogeneous data and make it open access so developers anywhere can train their predictive models on a unified data set.
They are also funding technology development centers, or TDCs, to build names for specific complex diseases like neurological disorders. And they created a validation and qualification network, the VQN, to help get these new tools through those FDA regulatory hurdles we just talked about.
It's a full pipeline approach.
But my absolute favorite part of the complementary announcement is the prize money.
The Reduction to Practice Challenge.
A $7 million cash prize for anyone who can develop a human-based M and turn it into a practical, working, commercially viable solution within 3 years. It operates like an XPRIZ for drug development.
It is entirely designed to push these technologies out of the academic labs and into the real-world industrial pipeline.
Because academic papers don't save lives, actual tools do.
Exactly. The goal isn't just a published paper. The goal is a tool that pharmaceutical companies can buy and use tomorrow.
We also have to look at the broader political context driving this federal funding. The Make America Health Again, or MAA, Commission released a strategy report in September 2025.
Right, that was a major catalyst.
That report explicitly targeted the inefficiencies of animal testing and resulted in the creation of 2 entirely new NIH offices.
They launched ORIVA, the Office of Research, Innovation, Validation, and Application, which is explicitly designed to scale NAMs and serve as the interagency coordination hub.
And the other one.
They launched ORIPA, the Office of Research, Innovations, Planning, and Analysis, to focus heavily on the chronic disease portfolio and improve scientific reproducibility across the board.
So what does this all mean? When you look at the FDA guidance, the $150 million complementary program, and these new NIH offices, it is a level of coordinated, top-down, interagency focus that is exceptionally rare.
It's unprecedented, really.
And the most shocking policy shift, the absolute hammer dropping on the old system, is what an NIH official announced at a recent joint FDA and NIH workshop.
This policy change forces the hand of the entire academic ecosystem. The NIH announced that moving forward, they will no longer fund grant proposals that exclusively rely on animal testing if viable NAMs exist for that specific research area.
Let me make sure I understand. If you want federal grant money, you cannot default to a mouse model just because it's what you've always done.
You cannot. Historically, scientists stuck with animal models simply because of decades of historical use.
It was the safe bet.
It was the safe, established way to get published and secure grant funding. You knew the peer reviewers understood mouse xenografts. But the NIH is removing that safety net. They are mandating adaptation to human-based methods.
Wow. So we've seen the incredible engineering, the regulatory shifts, and hundreds of millions in federal funding. Let's look at how this is actively changing specific fields of medicine right now. Let's look at oncology. Cancer research is notoriously difficult.
Oncology is perhaps the perfect case study for why NEMs are biologically essential. Cancer is incredibly complex, and our historical reliance on animal models in this field has been highly problematic, particularly with mouse xenografts.
Explain the biology of a mouse xenograft because it really highlights the flaw we discussed earlier.
In a standard xenograft model, researchers take a piece of a human tumor and graft it onto a mouse to test if a chemotherapy drug will shrink it.
Okay, sounds straightforward.
But to ensure the mouse's immune system doesn't immediately reject the foreign human tissue, researchers have to use ephemic nude mice that have been genetically engineered to have almost no immune system.
Wait, but the most advanced cancer therapies today, like Keytruda, are immunotherapies. They work by stimulating the patient's immune system to attack the cancer.
That is the structural mismatch. You cannot test a drug designed to stimulate the immune system in an animal model that fundamentally lacks an immune system.
That makes zero sense.
It doesn't. Furthermore, human tumors don't exist in a vacuum. Their behavior is dictated by the complex tumor microenvironment and vast genetic heterogeneity. A xenograft fails to mimic almost all of those human-specific complexities.
So that's why we see so many false positives in cancer research.
It's why we have successfully cured cancer in mice thousands of times, only to watch those same drugs fail in clinical trials.
How do N solve the cancer problem? The data points heavily to patient-derived organoids, or PDOs.
Patient-derived organoids are revolutionary for personalized medicine. Instead of putting a generalized tumor in a mouse, an oncologist takes a biopsy directly from a specific patient.
The actual person who is sick.
Yes. They then culture those cells in 3D in the lab, growing a miniaturized version of that exact patient's tumor.
So it retains the patient's unique biological architecture.
Crucially, it retains the patient's specific clonal architecture, genetic mutations, and tumor microenvironment. Doctors can take that organoid, split it up into dozens of microsamples, and test an entire panel of different chemotherapy drugs against it ex vivo, outside the patient's body.
So they don't have to guess which drug will work.
They can observe exactly which compound effectively kills that specific patient's tumor without having to expose the human patient to the toxic side effects of trial-and-error chemotherapy.
And they are combining these organoids with the microphysiological systems we talked about earlier to create cancer on a chip.
Cancer on a chip introduces blood flow and endothelial barriers to the organoid. You can circulate the patient's own immune cells through the microfluidic channels of the chip to see if a novel immunotherapy drug successfully helps the immune cells traffic into the tumor microenvironment and attack the cancer cells.
That is amazing.
You simply cannot model that dynamic immune trafficking in a standard mouse model.
And this isn't just theoretical. There is a very specific real-world success story we need to highlight. The FDA recently granted an investigational new drug, or IND, approval for a combination cancer therapy using solely NNAMs approaches.
It was a huge milestone for the industry. Developers combined an anti-cancer drug called BAL0891 with an immune checkpoint inhibitor called teslelizumab.
Okay.
They proved its efficacy and safety to the FDA using a highly advanced human vascularized organoid model. Absolutely no animal testing was required for that specific IND approval.
None at all.
None. The FDA evaluated the robust NAM data and determined it was fit for purpose to proceed to human trials.
That is the new map working in real time. Now, another interesting angle here is the industry perspective. In any massive technological shift, there is a foundational business story. It reminds me of how the internet was built. Like, the companies that reliably generate immense value aren't necessarily the ones building the flashy consumer websites. They are the companies building the physical servers, the fiber optic cables, the foundational infrastructure that allows the entire ecosystem to run.
In the NAMS revolution, companies like Thermo Fisher Scientific are building that foundational infrastructure.
Exactly. They aren't necessarily inventing the specific cancer drug. They are providing the raw materials that allow everyone else to build the NAMs.
The picks and shovels.
The picks and shovels of the gold rush. They supply the specialized primary human cells, the complex 3D growth media, the assay systems, the analytical software. They are providing the specialized Lego bricks for the entire global industry.
We can take that a step further by looking at their strategic advice. Because they are so deeply embedded in the supply chain of both traditional pharmaceutical development and the new NAMS ecosystem, their guidance to biotech companies is very pragmatic.
What do they advise?
They explicitly advise companies not to abandon animal testing completely overnight.
Which sounds completely counterintuitive given the 90% failure rate and the massive federal push we just talked about.
It sounds contradictory, but it is deeply practical. They advocate for a phased adoption. They advise companies to start small, running names alongside their traditional animal models in parallel.
To compare the data.
Exactly. This allows a company to build comparative internal evidence. You run the mouse toxicology test, and simultaneously you run the liver on a chip test. When the chip consistently predicts the human outcome better than the mouse over several years of trials, you build internal corporate confidence.
And data for the FDA.
And a bulletproof data package to build regulatory confidence. You gradually phase out the animals as the NAMs prove their absolute superiority in your specific drug pipeline.
You don't burn the old map until you are 100% sure the new GPS is locked on.
Yeah.
Which leads us perfectly into the final section of our deep dive. Because if this tech is so amazing, if it saves hundreds of millions of dollars per drug, and if it's ethically superior, why hasn't every single pharmaceutical company completely abandoned animal testing today? What are the bottlenecks? What is the reality check?
The reality check is that translating in vitro data to whole-body human physiology is incredibly difficult math, and the institutional roadblocks are massive. The first major bottleneck is cultural inertia.
Jacqueline Corrigan-Curie, the acting director of the Center for Drug Evaluation and Research at the FDA, touched on this cultural issue.
She did. She noted that large manufacturers default to animal testing simply because it is an established, heavily worn path. Big pharma is fundamentally risk-averse.
I mean, they're spending billions.
When a company is investing $1 billion into bringing a drug to market, they want to follow the path of least resistance to regulatory approval. For decades, that path was paved entirely with animal data.
So they are just scared.
There is a deep institutional fear of the unknown. Companies worry that if they submit a completely novel ANAM data package, an FDA reviewer might delay the drug simply because they aren't familiar with evaluating that specific methodology.
It's the old corporate mindset. Nobody ever got fired for buying IBM. In drug development, nobody ever got fired for submitting a standard rat toxicology study.
Exactly. Beyond the psychology, there's a very real, very daunting technical challenge that the industry is still actively wrestling with. It's an acronym called IVE.
IVE. In vitro to in vivo extrapolation. Wait, I'm stuck on the math here.
It's tricky.
If I test a drug on a tiny microfluidic liver chip that holds maybe a few thousand cells, how on earth do you mathematically translate that to a 200-pound human taking a 500-milligram pill? Like the absorption rates in the stomach, the blood volume, it all has to be wildly different.
That is the exact mathematical challenge of IVE. It relies on incredibly complex models called PBPK, physiologically based pharmacokinetic modeling.
Break that down for me.
Let's say you test a chemical on a liver on a chip, and you find that at a concentration of 10 micromolar, the liver cells begin to show toxicity. The IVVE challenge is translating that static microfluidic concentration into a real-world, whole-body dosing regimen.
So you have to calculate the entire journey of the pill.
Yes. You have to account for oral bioavailability, how much survives the stomach acid. You have to calculate the volume of distribution, how much diffuses into fat tissue versus staying in the blood. You have to calculate renal clearance, how fast do the kidneys filter it out.
And if your mathematical model is off by even a fraction, the dose prediction is completely wrong.
A study by Carlson et al. Perfectly highlights the danger of mathematical variance here. They demonstrated that depending on exactly which parameters you set in your PBPK model, like assuming slightly different protein binding affinities in the blood, the M's can predict a toxic human dose that is either exponentially higher or exponentially lower than traditional baseline estimates.
Wow. So if your math is slightly off, you might accidentally greenlight a highly toxic drug, or you might incorrectly kill a perfectly safe, life-saving cure just because the model said it was dangerous.
The variance is huge. That potential for variability makes regulators understandably nervous. This is why regulatory hesitation is still a massive bottleneck. They want to see bulletproof, highly standardized IVV models before they completely let go of the whole animal data they are used to evaluating.
To combat that specific hesitation, there is a strategy being deployed in Europe by the EMA, the European Medicines Agency. They've introduced the concept of a safe harbor.
The safe harbor approach is a brilliant policy mechanism for fostering innovation without risking public safety.
How does it work?
It allows drug developers to voluntarily submit their raw NM data to the regulators alongside their traditional animal data, completely outside the formal approval pathway, without any regulatory penalty.
Even if the NAM data contradicts the animal data or the math looks weird, the EMA won't reject the actual drug application based on that voluntary submission. It's like a sandbox.
It's a data sandbox. It allows the regulatory agencies to look at cutting-edge NA-mine data, learn from it, and figure out how to evaluate the complex IVAV math, all while protecting the innovator from being punished for trying something new. It builds mutual confidence between the regulators and the developers.
Okay, thinking about the biology here, let's say you nail the IV of V math perfectly, and your individual liver on a chip is 100% accurate. What about systemic multi-organ cascades?
The domino effect.
Right. Let's say a patient takes a new drug and it clears the liver perfectly, but the liver metabolizes that drug into a secondary chemical, and that metabolite travels up to the brain and causes a severe hormone imbalance. How does an isolated liver M catch a problem that only manifests in the brain?
This raises an important question, and it represents the absolute holy grail of current NAM engineering. An isolated liver chip cannot catch a brain issue. The solution and the future goal the industry is aggressively racing toward is integrating these isolated systems into interconnected human-on-a-chip models. A human on a chip, connecting the various Lego structures together with plumbing.
You physically link the microfluidic outflow of the gut chip to the inflow of the liver chip, and then link the metabolic exhaust of the liver chip to a kidney chip and a brain chip.
So it flows through them just like a real circulatory system.
You recreate whole-body systemic circulation, and you support all of that with advanced AI to monitor the complex pharmacokinetics of how a drug travels, metabolizes, and bioaccumulates through that interconnected microsystem.
Are we actually close to a functional, interconnected human on a chip? Because keeping all those vastly different organ tissues alive, perfectly oxygenated, and communicating in the same microfluidic system, that sounds like an absolute nightmare of engineering.
It is an incredible engineering hurdle, but we are making massive strides piece by piece. Look at the isolated components first. A recent evaluation of the Emulate liver chip demonstrated over 87% sensitivity and over 90% specificity in predicting drug-induced liver injury that traditional animal models had entirely missed.
That's already better than the animals.
Exactly. When you have individual organ components functioning at 90% accuracy, the next logical engineering step is coupling them together. It is no longer science fiction. It is an active, heavily funded engineering challenge.
That is just phenomenal, and it brings us full circle. We started this deep dive looking at a deeply flawed, incredibly expensive, and ethically fraught century-old paradigm of forcing human medicine through the structural filter of animal biology.
A filter that resulted in a staggering 90% clinical failure rate.
Right, and what we are witnessing now is the rapid, exceptionally well-funded assembly of a new map. A complex, interconnected box of human-centric technologies, living microphysiological chips, patient-derived organoids, and AI supercomputers processing multi-omics data.
The whole ecosystem is changing.
We've seen sweeping regulatory reform with the FDA Modernization Act 2.0 and the 2026 guidance, and massive capital injections from programs like the NIH's $150 million Complementary Initiative.
Regulators, federal funders, academic researchers, and massive biotech corporations, they are finally rowing in the exact same direction.
For anyone listening, whether you are managing a chronic illness, waiting for a breakthrough cancer treatment, or just picking up an over-the-counter medication, this matters because this revolution promises faster drug development, significantly cheaper medications due to the reduction of billion-dollar failure rates, and most importantly, treatments that are actually engineered for your human biology, not a macaque's biology.
Before we wrap up, I want to leave you with one final provocative thought regarding where this technology is heading.
Let's hear it.
As AI, multi-omics, and these microphysiological systems advance and integrate, we are rapidly moving toward the creation of highly personalized digital twins.
Digital twins, meaning a complete computational and biological model of a single specific person.
In the near future, we won't just be skipping generalized animal testing. If you are diagnosed with a complex disease, your doctor might take a biopsy, grow your specific organoid on a chip, and run millions of toxicological simulations on your computational digital twin.
Just yours.
Just yours. They will know exactly which drug will cure you and exactly which drug will cause you a severe side effect, based entirely on your unique genomic and proteomic profile before a single drop of medicine ever enters your actual body.
It's the end of the average human clinical trial entirely.
It raises the ultimate question. Are we currently witnessing the end of trial and error medicine as we know it?
Suddenly, that diagnostic and developmental landscape we talked about at the beginning doesn't look so muddy anymore. The picture is getting crystal clear. Thank you for joining us on this deep dive into the future of medicine. Keep exploring, keep questioning, and we'll see you next time.